Imagine standing at the edge of the ocean, watching waves roll endlessly toward the shore. The water looks empty, doesn't it? Clear blue stretching to the horizon, perhaps a few jellyfish drifting near the surface, maybe a school of fish flashing silver in the sunlight. But what if I told you that every liter of that seemingly vacant water contains hundreds, sometimes thousands, of living creatures? Not bacteria or plankton, but the infant forms of the ocean's most familiar inhabitants. Crabs that haven't yet grown their shells. Fish that look nothing like their parents. Starfish without arms. They float there, translucent as glass, nearly invisible to human eyes. These are the larvae, the children of the currents, and they outnumber every whale, every shark, every coral polyp by billions to one. This is their ocean, not ours. Scientists estimate that 70% of all marine species spend their earliest days or weeks in this drifting larval form. That percentage climbs even higher when you consider the sheer abundance of individuals, because a single fish might lay millions of eggs, each hatching into a larva no bigger than a grain of sand. These microscopic wanderers travel distances that would be impossible for their adult forms. Cod larvae drift from offshore spawning grounds in the North Sea all the way into coastal Norwegian fjords, covering hundreds of kilometers in their first months of life. Deep-sea vent species somehow traverse 20 miles along mid-ocean ridges, navigating between volcanic islands of life separated by barren seafloor. Brittle star larvae can drift on deep currents for so long that populations separated by entire ocean basins remain genetically connected. How does a creature with barely any nervous system accomplish journeys that human navigators once needed stars and compasses to achieve? For over a century, marine biologists assumed larvae were passive drifters, helpless specks carried randomly by currents like dust in the wind. The mathematics seem to support this. Ocean currents are powerful, moving at speeds that would sweep away anything so small and weak. Adult fish can barely swim against strong currents, so how could their larvae, thousands of times smaller and with primitive muscles, exert any control over their fate? The prevailing theory held that survival was purely a numbers game. Spawn millions of larvae, and a few lucky ones would randomly drift to suitable habitats. The unlucky majority would starve, be eaten, or wash up in environments where they could never survive. This was ocean ecology as lottery, existence as cosmic accident. But then scientists started looking closer, and what they found didn't match the theory at all. Larvae weren't drifting randomly. They were arriving at specific destinations with impossible consistency. Herring larvae in Norwegian waters showed elevated recruitment success when they relocated rapidly northward toward the Barents Sea, as if they knew where they were going. When currents failed to carry them in the right direction, recruitment collapsed. Larvae were somehow sensing their environment and responding to it. Researchers discovered that many species perform precise vertical migrations, swimming up toward the surface at night and descending deeper during the day, riding different current layers like elevators between floors. Some larvae could detect chemical signals in the water, following scent trails back to their natal reefs from distances of many kilometers. Others appeared to sense magnetic fields, orient themselves using polarized light, or respond to pressure changes that indicated proximity to coastlines. These weren't reflexes. These were navigation strategies. But here's where the mystery deepens into something almost unsettling. Many of these larvae have nervous systems so primitive that calling them brains feels generous. We're talking about clusters of neurons you could count on your fingers. Some larvae are little more than swimming digestive tracts with light-sensitive patches and balance organs. Yet they're making decisions. A larval fish might remain in surface waters traveling one direction, then suddenly dive 50 meters to catch a deeper current flowing the opposite way. Why? How does it know to do that? What is it sensing that tells it this current leads to survival and that one leads to death? Scientists have built underwater robots that mimic larval behavior, programming them with the same swimming patterns and sensory responses observed in real larvae. These robot larvae can successfully navigate to settlement sites. But the robots are following our instructions, executing algorithms we designed. The real larvae have no programmer, no external guidance. So who taught them? Consider the scale of what we're really discussing. At any given moment, trillions upon trillions of larvae populate the world's oceans, each one navigating according to strategies encoded in its genes and refined through sensory feedback from its environment. They form an invisible web of life in constant motion, connecting ecosystems across thousands of kilometers. Every coral reef depends on larvae arriving from distant reefs to replenish its population. Every coastal fishery relies on larvae successfully completing their journey from offshore spawning grounds. The entire architecture of ocean biodiversity rests on the success of these nearly microscopic travelers. Remove larval transport and marine ecosystems collapse within a generation. Yet we didn't even know this invisible foundation existed until embarrassingly recently. We were studying the ocean while being completely blind to the mechanism that literally holds it together. What makes this even more fascinating, more mysterious, is that we still can't fully explain how the system works. Yes, we've identified some sensory mechanisms. Yes, we can track larvae using sophisticated modeling and genetic analysis. But the fundamental question remains stubbornly unanswered. How does a creature with such limited cognitive capacity execute such complex behaviors? Is there something we don't understand about consciousness, about intelligence, about the minimum requirements for decision-making? Some researchers have begun suggesting that larvae might exhibit a form of distributed intelligence, that the swarm itself processes information in ways no individual larva could. Others point to chemical communication between larvae, suggesting they coordinate their movements through signals we haven't yet learned to detect. The ocean might be humming with larval conversations we simply can't hear. So the next time you stand at the ocean's edge, looking out at what appears to be empty water, remember what's really there. Remember the billions of invisible travelers riding currents you can't see, navigating by senses you don't possess, toward destinations they've never visited, but somehow know how to find. Remember that these creatures, these children of the currents, are not just drifting. They're not just surviving. They're accomplishing something far stranger and more sophisticated than passive existence. They're writing the future of the ocean with every choice they make, every current they ride, every chemical signal they follow through the darkness. And we're only beginning to suspect how much they know, how much they control, and what happens when their invisible journeys start leading them somewhere new. Something is changing in the larval realm, and we need to understand what before those changes reshape every ocean ecosystem on Earth. If you wanted to design the perfect navigator, what would you give it? Perhaps an advanced brain capable of processing complex spatial information. Sharp eyes to read the stars or identify landmarks. Maybe sophisticated instruments to measure magnetic fields, water chemistry, temperature gradients. Now imagine stripping all that away until you're left with something barely more complex than a grain of rice with a few nerve cells and some sensory patches. This is the navigator we're actually dealing with when we talk about marine larvae. Yet somehow, against all reasonable expectations, these microscopic creatures accomplish navigational feats that rival or exceed the capabilities of much larger, supposedly more intelligent animals. A sea turtle hatchling that returns to its natal beach decades later gets celebrated as a marvel of nature. But that turtle has a real brain, functional eyes, and years of experience. A larval fish accomplishes similar precision in its first weeks of life with neural equipment that wouldn't impress a earthworm. The evidence for active larval navigation has been accumulating for decades, but only recently has the full scope of their abilities become clear. Coral larvae can detect and orient toward calcium ions dissolved in seawater, following concentration gradients back to reef structures from distances of several kilometers. When researchers created artificial chemical gradients in laboratory channels, larvae didn't just drift passively. They actively swam toward calcium sources while avoiding magnesium-rich areas. Even more remarkably, they changed their swimming patterns based on chemical concentration. In high-calcium zones, larvae increased their turning behavior, essentially executing a local search pattern as if they knew they'd arrived somewhere important. Near crustose coralline algae, which produces chemical exudates that signal suitable settlement habitat, larvae exhibited similar searching behaviors. They were reading the water like a map, interpreting chemical signals we can barely detect with sophisticated instruments. But chemical sensing is just one tool in the larval navigation toolkit. Many species perform elaborate vertical migrations that suggest they're deliberately choosing which currents to ride. During daylight hours, These larvae descend deeper into the water column where currents might flow in one direction. At night, they rise toward the surface where different currents carry them elsewhere. By timing these vertical movements, larvae can effectively steer themselves across horizontal distances despite having swimming speeds that seem pathetically inadequate compared to ocean currents. Think of it like crossing a river. You can't swim faster than the current, but by angling your body and choosing where to enter the water, you can control where you exit on the opposite bank. Larvae do this in 3 dimensions, continuously adjusting their depth to exploit favorable currents while avoiding unfavorable ones. Some species maintain this pattern for weeks, making hundreds of vertical migrations totaling distances of several kilometers up and down through the water column. Then there's the magnetic sense. Several species of larval fish appear capable of detecting Earth's magnetic field and using it for orientation, much like migratory birds. In laboratory experiments, larvae exposed to artificial magnetic fields altered their swimming directions in ways that suggested they were attempting to maintain a preferred compass heading. This makes evolutionary sense because magnetic field lines provide reliable directional information across vast oceanic distances where visual landmarks don't exist and chemical signals become too diluted to follow. But here's the unsettling part. The sensory organs and neural circuitry required for magnetoreception in birds are already considered mysterious, poorly understood compared to vision or hearing. How larvae accomplish the same feat with vastly simpler biology remains almost completely unknown. We know they're doing it. We just can't explain how. Perhaps the most inexplicable phenomenon is synchronized metamorphosis. In species after species, researchers have documented events where thousands or millions of larvae settle and transform into their adult forms simultaneously, often over the course of a single night. These mass settlement events aren't triggered by simple environmental cues like water temperature or lunar phase. Larvae from the same cohort can be separated by kilometers of ocean, exposed to slightly different local conditions, yet they still metamorphose in tight synchronization. It's as if they're communicating with each other across distances, coordinating their life history transitions. Some scientists have proposed that larvae release chemical signals that propagate through the water, creating feedback loops where early settlers trigger settlement in nearby larvae, which triggers more settlement in an expanding cascade. But this doesn't explain synchronization across large distances where chemical signals would dissipate. Others have suggested that larvae might respond to the same subtle environmental cues in coordinated ways, essentially reading shared information from their environment. But what cues? What information? This brings us to one of the most fascinating and frankly disturbing hypotheses in marine biology. What if individual larvae aren't really making these decisions at all? What if the swarm itself is the functional unit of intelligence? In distributed intelligence systems, individual agents follow simple rules based on local information, but the collective behavior emerges at a higher level of organization. Ant colonies work this way. No individual ant understands the colony's architecture, but together they build complex structures. Starling murmurations create elaborate aerial displays without any bird serving as a leader. Could larval swarms operate on similar principles? Each larva might respond to simple stimuli like light intensity, chemical concentrations, or the presence of other larvae. But when billions of larvae interact through these simple responses, complex navigation strategies and coordinated behaviors might emerge from the collective without any individual larva understanding what it's doing. The implications of this possibility are profound and more than a little eerie. It would mean the ocean is humming with an intelligence we've never recognized, a form of awareness distributed across billions of nearly invisible bodies rather than concentrated in individual brains. When we observe a larval swarm successfully navigating to distant reefs or coordinating their settlement timing, we might be witnessing something closer to a single organism thinking rather than a crowd of individuals acting independently. And if that's true, what happens when we disrupt these swarms? When pollution kills off portions of a larval cohort, are we just reducing population numbers, or are we lobotomizing a distributed intelligence, removing critical nodes from a network that thinks across space and time? When ocean acidification alters the chemical signals larvae use to communicate, are we simply adding noise to their environment, or are we scrambling their language, making it impossible for the collective to form coherent thoughts? This is where the mystery deepens into something approaching horror. because larvae aren't navigating through a stable environment anymore. Ocean currents are shifting as climate patterns change. Chemical signals are being altered by acidification and pollution. Temperature gradients that once provided reliable navigational cues are becoming unpredictable. Magnetic field anomalies are spreading across migration routes. And the larvae are noticing. Settlement patterns are changing. Species are arriving in ecosystems where they've never been recorded. Some larvae appear to be delaying metamorphosis, remaining in larval form far longer than their biology should allow, as if they're waiting for signals that never come. Others are settling prematurely in unsuitable habitats, transforming into juveniles that cannot survive. The navigation system that has reliably delivered larvae to appropriate habitats for millions of years is breaking down. And we don't know if larvae can adapt fast enough. We don't know what happens when phantom navigators lose their way. But we're about to find out whether we're ready for the answer or not. We tend to think of larvae as vulnerable, helpless things. Tiny morsels adrift in a predator-filled ocean, surviving only through sheer numbers and luck. This image is comforting because it casts larvae as passive victims in the marine food web, occupying their proper place at the bottom of the hierarchy. But this comfortable narrative is wrong. Many larvae are not defenseless at all. They're armed with chemical arsenals that would impress a military weapons designer. Some produce toxins more potent than their adult forms ever will. Others hijack the bodies of larger organisms, turning them into unwilling transportation and protection. A few species have evolved chemical defenses so sophisticated that they can selectively poison predators while leaving other species unharmed. This isn't passive drifting. This is chemical warfare at a microscopic scale, and it's been raging in the oceans for hundreds of millions of years. We're only now beginning to understand how brutal and complex this invisible war really is. Take the larvae of certain ascidians, those bizarre filter-feeding tunicates that cover underwater surfaces like translucent grapes. Their larvae look harmless enough, resembling microscopic tadpoles with simple eye spots and a primitive notochord. But chemical analysis reveals they're saturated with compounds called didemnins, cyclic peptides that bind to proteins involved in cell division and literally stop cells from reproducing. These aren't trace amounts we're talking about. Larval tissue contains didemnin concentrations high enough that a single larva, if consumed, causes immediate regurgitation in predatory fish. The fish learns quickly. After one encounter, it will avoid larvae with that particular shape and color pattern for weeks. This is Pavlovian conditioning weaponized at the larval stage. The larvae are teaching their predators to fear them, one nauseating lesson at a time. And here's the disturbing part. Adult ascidians often contain far lower concentrations of these same compounds. The larvae are more toxic than their parents. Evolution has invested the most deadly chemistry in the life stage that appears most vulnerable. But producing your own toxins is expensive metabolically. It requires genetic machinery to synthesize complex molecules and energy to manufacture them in meaningful quantities. Some larvae have found a more efficient solution. They steal their chemical weapons from other organisms. Nudibranch larvae, those colorful sea slug juveniles, inherit defensive compounds called tyriotoxins and other toxins from their parents, who sequester these chemicals from the sponges and cnidarians they consume. The parents transfer toxins directly into their eggs, essentially arming their offspring before they're even born. When those larvae hatch, they're already protected by poisons manufactured by completely different organisms, sometimes across multiple degrees of ecological separation. A coral produces nematocysts containing toxins. A nudibranch eats the coral and stores the toxins without being harmed. The nudibranch lays eggs infused with those toxins. The larvae inherit protection from a predator-prey relationship they've never directly participated in. This chemical defense as inheritance, toxicity as birthright. Then we have the parasites, and this is where larval strategy crosses from clever into genuinely horrifying. Barnacle larvae of the species Saculina don't settle on rocks like their normal cousins. They hunt for crabs. When a larval Saculina finds a suitable crab host, it doesn't simply attach to the surface. It injects itself into the crab's body, discarding most of its own anatomy in the process. What remains spreads through the crab's tissues like fungal threads, infiltrating every organ system. The parasite doesn't kill its host. That would be wasteful. Instead, it chemically castrates the crab, hijacks its reproductive system, and forces it to care for the parasite's eggs as if they were the crab's own offspring. The crab will fan and clean the parasite's egg mass, protect it from predators, and eventually disperse the next generation of Sacculina larvae into the water. The host has become a puppet, its behavior rewritten by parasitic chemistry. And this happens to millions of crabs right now in oceans around the world. Parasitic larvae have evolved strategies that seem almost impossible without intelligence guiding them. The larvae of certain trematode flatworms infect snails as their first host, but they need to get into a bird to complete their life cycle. So they migrate to the snail's eye stalks, forming bright-colored cysts that pulse rhythmically. The infected snail becomes conspicuous, easy prey for birds that would normally avoid healthy snails. The larvae have transformed their host into bait, advertising its location to the next stage in their parasitic journey. Other larvae manipulate host behavior by disrupting neurotransmitter production, essentially rewiring their victims' brains with chemical precision. How does a larvae with almost no nervous system know which neurotransmitters to target? How did evolution discover these specific molecular keys that unlock behavioral manipulation? The mechanisms are so precise they suggest millions of years of trial and error, countless failed attempts, and successful strategies refined through incomprehensible spans of time. But perhaps the most unsettling aspect of larval chemical warfare isn't individual defense or parasitism. It's what happens when defensive larvae appear in overwhelming numbers. These events are called phantom blooms, and they're exactly as ominous as they sound. Under certain conditions, billions of chemically defended larvae can concentrate in specific areas, creating toxic zones that kill fish, invertebrates, and even marine mammals. The larvae themselves aren't deliberately coordinating an attack. They're simply following normal behavioral patterns, aggregating in response to currents, food availability, or settlement cues. But the cumulative effect of their chemical defenses can poison entire bays. Fish swimming through these zones experience respiratory distress, disorientation, and sometimes death. Filter feeders that consume the larvae die from toxin accumulation. The water itself becomes temporarily uninhabitable, a no-go zone created by organisms that individually weigh less than a grain of sand. What makes phantom blooms particularly terrifying is how little we understand about what triggers them. They seem to appear without warning, persist for days or weeks, and then disperse as mysteriously as they formed. Some researchers suspect that climate change is increasing bloom frequency by altering larval development timing and current patterns. Warmer waters might accelerate larval development, causing multiple species to release their larvae simultaneously rather than in staggered pulses across several weeks. This temporal compression could create superblooms where chemical defenses from dozens of species combine in unpredictable ways. We're also seeing blooms in areas where they've never been recorded, suggesting that larval distributions are shifting as ocean conditions change. Are we witnessing the beginning of a new pattern? Will phantom blooms become routine rather than exceptional? No one knows. We're observing the phenomenon in real time without understanding its full implications. The chemical war happening in the larval realm isn't static. It's an evolutionary arms race that never stops, never reaches equilibrium. As predators evolve resistance to larval toxins, larvae evolve more potent compounds or entirely new chemical strategies. As settlement substrates develop anti-fouling chemicals to prevent larvae from attaching, larvae evolve mechanisms to detect and avoid those chemicals, or they develop tolerance and settle anyway. Each adaptation triggers counter-adaptations in an endless cycle of chemical innovation. And now climate change has thrown an accelerant onto this ancient conflict. Warming waters, ocean acidification, and pollution are changing the rules of engagement. Some chemical defenses become less effective in acidic water because the compounds degrade faster or predators develop higher tolerance under stress. Other defenses become more potent as warmer temperatures increase metabolic rates and toxin production. We're not just observing this arms race anymore. We've become participants, whether we intended to or not. And the larvae, those invisible warriors armed with molecular weapons, are adapting faster than we can track. The chemical war is escalating, and we're only beginning to realize we're caught in the crossfire. Evolution is supposed to be slow. We learn about natural selection as a process requiring thousands of generations, geological timescales, changes accumulating so gradually that no human lifetime could witness them directly. Darwin's finches needed millennia to diversify their beaks across the Galapagos Islands. Peppered moths took decades to shift from light to dark coloration during the Industrial Revolution, and we celebrated that as remarkably fast evolution. But what if I told you that some marine larvae are evolving measurably in just a few years? Not adapting through behavioral flexibility or phenotypic plasticity, but actually changing their genetic makeup, selecting for new traits, becoming functionally different organisms faster than we can study them. This is evolution in fast forward, driven by environmental changes so rapid and severe that only the most extreme variants survive to reproduce. And it's happening right now in oceans around the world, largely invisible to us because the organisms involved are microscopic and the environments they inhabit are vast and difficult to monitor. We're witnessing evolutionary change at a pace that shouldn't be possible, and the implications are as fascinating as they are terrifying. The mechanism is actually straightforward once you understand larval biology. Most marine species produce enormous numbers of offspring, millions of larvae from a single spawning event. Within that massive cohort exists tremendous genetic variation. Some larvae develop slightly faster, others slightly slower. Some tolerate warmer temperatures, others prefer cooler water. Some can function in more acidic conditions, others cannot. Under stable environmental conditions, this variation doesn't matter much. The range of phenotypes all fall within acceptable parameters, and survival rates across the genetic spectrum remain relatively similar. But when environmental conditions shift dramatically, suddenly that variation becomes life or death. Larvae at the extreme edges of the tolerance spectrum, individuals that would have been merely average in a stable environment, become the only survivors. They're not better larvae in any absolute sense. They're just better suited to the new, altered conditions. And when only they survive to reproduce, their genes dominate the next generation. This is the larval bottleneck, and climate change is forcing species through it at unprecedented speed. Consider what's happening with coral larvae right now. Corals spawn once per year in massive synchronized events, releasing billions of larvae into water that's progressively warming and acidifying. Most coral larvae have narrow thermal tolerance windows. A temperature increase of just 2 or 3 degrees Celsius during their vulnerable early development can cause catastrophic mortality. In the past, occasional warm years would kill off temperature-sensitive variants, but cooler years would follow, allowing the full range of genetic diversity to persist in the population. But now we're seeing consecutive warm years, sometimes entire decades where ocean temperatures never return to historical norms. This sustained pressure eliminates not just the most temperature-sensitive larvae but also the moderately sensitive ones. Only larvae at the absolute extreme of heat tolerance survive. When they settle and grow into adult corals, they spawn larvae that inherit those extreme tolerances. Within just a few generations, we're observing coral populations with measurably different thermal thresholds than their ancestors had 20 years ago. The species hasn't changed, but the population has been genetically filtered through repeated larval bottlenecks. What makes this process genuinely unsettling is how quickly it can produce outcomes that seem impossible. Research on fish larvae from warming waters shows some populations developing faster at higher temperatures while simultaneously requiring less food. Metabolic changes that shouldn't be compatible according to standard physiological models. Higher temperatures normally increase metabolic rates, which should increase food requirements, not decrease them. Yet these larvae are somehow doing more with less, operating under rules that violate what we thought were fundamental constraints. Are they accessing energy reserves through novel biochemical pathways? Have they altered their cellular machinery to be more efficient? We don't know yet. We just know that larvae from warm-adapted populations survive and grow in conditions that kill their ancestors, and they do it using biology we don't fully understand. Evolution isn't just selecting for heat tolerance. It's producing genuinely novel physiological solutions that didn't exist in the parental gene pool, emerging through combinations of genetic variants that were never tested together before. Then there are the larvae that refuse to metamorphose. Every larval species has a programmed development timeline, a genetically determined window during which metamorphosis should occur. Larvae detect environmental cues indicating suitable settlement habitat, water temperature, specific chemical signals, and when conditions align, transformation begins. But in populations experiencing rapid environmental change, researchers are documenting larvae that delay metamorphosis far beyond their normal timeline. Some extend their larval duration by 50% or more, remaining in planktonic form while they continue drifting, searching for settlement cues that match their genetic programming. Others appear to enter a kind of developmental stasis, neither growing nor transforming, just surviving in a holding pattern. A few species show larvae that can extend their planktonic period almost indefinitely, creating what amount to eternal juveniles, organisms trapped between life stages because the environmental conditions that trigger normal development no longer exist. What happens to these delayed larvae? Do they eventually settle in marginal habitats? Do they die without ever completing metamorphosis? Or are we creating a new category of marine life, perpetual larvae that have given up waiting for a suitable future? The opposite phenomenon is equally disturbing. Some larvae are metamorphosing prematurely, transforming into juveniles before they've reached appropriate developmental stages or arrived at suitable habitats. This appears to be a stress response, a biological panic button triggered when larvae detect environmental conditions deteriorating beyond survivable limits. If the water is becoming too warm, too acidic, too depleted of oxygen, remaining in larval form guarantees death. Premature metamorphosis at least offers a chance, however slim, of survival. So larvae settle wherever they happen to be, transforming on substrates that cannot support their adult form, in water depths that don't match their physiological requirements, in ecosystems where they have no suitable food sources. The results are predictable and heartbreaking. Juvenile fish settle in seagrass beds meant for different species and starve within days. Coral larvae metamorphose on bare rock without the bacterial communities they need to complete development. Crab larvae transform before their carapaces have properly formed, emerging as soft, defenseless juveniles immediately consumed by predators. These aren't successful adaptations. These are desperate gambles that almost never pay off. But they're happening with increasing frequency as larvae run out of options. What should terrify us is that these responses are heritable. Larvae that delay metamorphosis produce offspring that also delay metamorphosis, often with even greater delays. The panic response becomes encoded in the genetic line, passed down as a permanent trait rather than a temporary stress reaction. We're not just watching individual larvae fail to adapt. We're watching entire populations genetically commit to strategies that might be catastrophically maladaptive in the long term. A population that evolves to tolerate extremely high temperatures might lose its ability to function in cooler water, effectively trapping itself in a thermal niche that's still shifting. A population that evolves to delay metamorphosis indefinitely might lose the ability to settle at all, producing larvae that drift until they die of old age or starvation. Evolution doesn't optimize for long-term survival. It optimizes for immediate reproductive success. And in rapidly changing environments, what works right now might doom future generations to extinction. The larvae surviving these bottlenecks aren't necessarily the ones that will build healthy ecosystems in the future. They're just the ones that didn't die yet. Perhaps most disturbing is the realization that we can't predict where this is heading. Traditional evolutionary models assume that environmental change occurs slowly enough for populations to explore the full range of adaptive solutions, testing different genetic combinations across many generations until optimal strategies emerge. But rapid change doesn't allow that exploratory process. Populations get locked into whatever solutions happen to exist in their gene pool at the moment selection pressure hits. If the right genetic variants aren't present when the bottleneck occurs, the population goes extinct regardless of how much potential for adaptation existed in their evolutionary history. We're essentially forcing species through evolutionary gauntlets without knowing if they have the genetic tools to survive. Some will make it, many won't. And the ones that do survive will be fundamentally different organisms than their ancestors, adapted to ocean conditions that have never existed in Earth's history. We're not just warming the ocean. We're rewriting the genetic code of larval life, selecting for traits and physiological strategies that evolution would never have discovered under natural conditions. The children of the currents are becoming something new, something unknown, and they're carrying those changes with them wherever the currents flow next, spreading adaptations born from climate chaos into every corner of the ocean. Evolution in fast forward doesn't give us better organisms. It gives us desperate survivors shaped by crisis, and we have no idea what kind of ecosystems they'll build when they finally settle and start reproducing. We're about to find out whether we're ready or not. Every ecosystem on Earth was built by invaders. That coral reef off the coast of Australia? 10,000 years ago it was bare rock, colonized by larvae drifting from distant reefs until enough species arrived and established to create the complex community we see today. That kelp forest in California, assembled piece by piece as larvae of kelp, fish, urchins, and countless other species found their way there through oceanic currents. Invasion is the ocean's natural building process, the mechanism by which life spreads across the planet. But there's a crucial difference between natural colonization and what's happening now. Natural invasion operates on geological timescales, giving ecosystems centuries or millennia to absorb newcomers, to develop relationships, to reach some kind of equilibrium. The invasions we're witnessing today are happening at unprecedented speed, driven by human transportation networks and accelerated by climate change. We've created larval superhighways that connect ecosystems that were never meant to be connected, and we've done it so quickly that receiving ecosystems have no time to adapt. The children of the currents are using our infrastructure to go places they could never have reached naturally, and they're bringing with them all the evolutionary adaptations we discussed before. Chemical weapons, rapid evolution, desperate survival strategies. And they're deploying these tools in ecosystems that have never encountered them before. The mechanism is actually quite simple, almost elegant in its efficiency. A cargo ship takes on ballast water in one port to stabilize itself, then releases that water in another port thousands of kilometers away. Within that ballast water are billions of larvae, eggs, and microscopic organisms, many of which survive the journey in the ship's tanks. When the ballast is discharged, these travelers suddenly find themselves in completely foreign waters. Most die immediately, unable to cope with different temperatures, salinities, or chemical conditions. But remember the larval bottleneck we discussed. A tiny percentage of that massive number will have genetic variants that allow them to tolerate the new conditions. And because they're larvae, they arrive precisely at the life stage best suited for establishing new populations. They're designed to settle and metamorphose in unfamiliar environments. That's literally their evolutionary purpose. We've just given them the transportation to reach environments that natural currents never could have delivered them to. It's as if we've invented teleportation for organisms that were already expert colonizers. The combination is devastatingly effective. Consider the numbers. Global shipping moves approximately 11 billion tons of ballast water around the world every year. That's 11 billion tons of ocean water containing trillions upon trillions of larvae, being mixed and redistributed across ecosystems that evolved in isolation for millions of years. A single large cargo vessel can carry up to 60,000 tons of ballast water, potentially containing thousands of species in their larval forms. Most of these ships travel routes that wouldn't exist in nature, connecting temperate ports with tropical ones, mixing Pacific communities with Atlantic ones, bridging northern and southern hemispheres. We've created an artificial circulatory system for the ocean, but instead of maintaining balance, it's scrambling genetic material and species distributions faster than evolution can respond. And this is just ballast water. We haven't even discussed larvae attaching to ship hulls, hiding in seafood shipments, or riding currents altered by coastal development and climate change. The invasions themselves follow predictable but horrifying patterns. First comes the ghost phase, when larvae arrive and settle but their populations remain too small to detect. They're there, growing, reproducing at low levels, but marine surveys miss them because they're rare and often mistaken for native species or simply overlooked. This phase can last years or even decades. Then comes the establishment phase, when populations reach critical mass and suddenly become visible. A few individuals turn into hundreds, then thousands, then millions. By the time we notice them, it's already too late. The third phase is expansion, where the established population begins producing its own larvae, spreading from the initial invasion site to nearby ecosystems. This is where the real damage occurs because these aren't just foreign species anymore. Remember, they've been filtered through survival bottlenecks. They're the variants that could tolerate the new environment, often making them more resilient than native species that evolved under stable conditions. The final phase is ecosystem transformation, where the invader becomes so abundant that it fundamentally alters how the ecosystem functions. Native species get displaced. Food webs reorganize. The entire character of the ecosystem changes. Let me give you a concrete example that's unfolding right now. The European green crab has invaded coastlines on 5 continents via larval transport in ship ballast water and deliberate introductions for culinary purposes. But here's where it gets interesting. The populations invading different regions aren't identical. The crabs that established on the west coast of North America came from European populations adapted to colder northern waters. As they spread southward down the coast, they encountered progressively warmer conditions. Traditional invasion theory would predict their range would be limited by thermal tolerance. Instead, we're watching them adapt in real time. Populations at the southern edge of their range are producing larvae with measurably higher heat tolerance than their parents just a few generations back. These heat-adapted larvae are being carried even farther south by coastal currents, establishing populations in waters that should have been too warm. The invasion front keeps advancing because the larvae are evolving faster than the environment can filter them out. They're not just invading space. They're invading climatic zones that were supposed to be barriers. The truly terrifying efficiency of larval invasion becomes clear when you map it over time. Scientists have tracked the spread of certain invasive species using genetic analysis of settled populations combined with larval dispersal models. What they've found is that once an invasive species establishes a foothold, its larvae can colonize suitable habitat up to several hundred kilometers away within a single generation. That bridgehead population then produces its own larvae, extending the range another few hundred kilometers. The expansion proceeds as a wave, moving outward from the initial invasion site at speeds determined by current patterns and larval development time. For species with long larval durations and favorable currents, this can mean range expansions of thousands of kilometers per decade. The invasive Ascidian Didymnum vexillum, a colonial tunicate, spread across the northwest Atlantic coast at rates exceeding 100 kilometers per year, carpeting docks, boat hulls, and seafloor habitat with its dense mats. Native species that took millennia to colonize the same coastline after the last ice age were displaced in just a few decades by an invader hitching rides on boats and spreading via larvae that could survive in ballast tanks. What makes these invasions particularly insidious is how they interact with climate change. As oceans warm, thermal barriers that once limited species distributions are disappearing. Temperate species are extending their ranges poleward, following the shifting isotherms toward cooler water. But they're not just drifting slowly northward. They're leapfrogging, with larvae dispersing hundreds of kilometers in single jumps, landing in newly suitable habitat that was too cold just decades before. Meanwhile, tropical species are expanding into what were formerly temperate zones, bringing with them entire assemblages of associated organisms. Coral larvae are settling on reefs farther from the equator than they've been in recorded history. Fish larvae originally confined to subtropical waters are recruiting into temperate ecosystems. The biogeographic zones that organized ocean life for millions of years are dissolving, replaced by novel communities assembled from species that never coexisted before. And the glue holding these new communities together is larval transport, that invisible network of drifting juveniles connecting distant populations and carrying evolutionary innovations across vast distances. Here's the deeply unsettling realization we need to confront. We can't stop this. Even if we eliminated ballast water discharge tomorrow, even if we halted all maritime shipping and eliminated every pathway of human-mediated transport, climate change has already altered ocean currents and thermal regimes enough that natural larval dispersal will continue redistributing species. The larvae don't need our ships anymore. We've warmed the water enough that thermal barriers have fallen. We've acidified the ocean enough that chemical signals are scrambled. We've altered current patterns enough that larvae are reaching places they couldn't before. The invasions will continue because we've fundamentally changed the rules governing where larvae can survive and how they move through the ocean. Every one of those evolutionary adaptations we discussed, the heat tolerance, the chemical weapons, the accelerated development, those traits are now propagating through larval dispersal networks at global scales. We haven't just enabled invasions. We've weaponized the ocean's natural colonization process, turned it into something faster, more aggressive, and more destabilizing than anything that's occurred in recent evolutionary history. The children of the currents have become an invasion force, and they're rewriting the biogeography of the ocean whether we intervene or not. But perhaps, just perhaps, we can learn to work with this process rather than fighting it. Because the same mechanisms driving these invasions once built every healthy ecosystem that exists. We need to understand how. What if everything we've discussed, all the mystery and horror, the invasions and adaptations, the chemical warfare and rapid evolution, what if none of it is actually a crisis? What if it's exactly what the ocean has been doing for billions of years, just accelerated and made visible by the scale of human impact? Every ecosystem we've called pristine was once empty rock colonized by larvae. Every stable community we've tried to protect is actually a snapshot of a process that never stops moving, never reaches equilibrium, always rebuilding itself through larval transport and settlement. The coral reefs we romanticize as ancient and unchanging are perhaps 5,000 years old at most, assembled by larvae during the relatively stable Holocene climate period. Before that, different reefs existed in different locations, built by different species adapting to different conditions. The ocean doesn't preserve, transforms. And larvae are the mechanism of transformation, the architects constantly redesigning ecosystems in response to changing conditions. We've been treating larval disruption as ecosystem collapse when it might actually be ecosystem adaptation. The children of the currents aren't destroying the ocean's future. They're writing it in real time using the only tools evolution has given them. And perhaps, if we can learn their language, we can participate in that writing process rather than just watching in horror. Scientists are already beginning to test this hypothesis through coral restoration projects that work with larvae rather than against them. Traditional coral restoration involved growing coral fragments in nurseries and manually transplanting them to degraded reefs, a labor-intensive process that could restore perhaps a few hundred square meters of reef per year at enormous cost. But newer approaches focus on larval seeding, essentially helping larvae do what they already know how to do. Researchers collect coral spawn during mass spawning events, rear the larvae through their most vulnerable early stages in protected conditions, then release millions of competent larvae directly onto degraded reef substrate. The larvae settle, metamorphose, and begin growing in densities far higher than natural recruitment could achieve. Within a few years, these seeded reefs develop into functioning ecosystems, complete with the fish, invertebrates, and other species that naturally recruit to healthy reef habitat. The success rate isn't perfect. Many larvae still die, but the scale is transformative. A single seeding event can restore hectares of reef, and the cost per square meter is orders of magnitude lower than fragment transplantation. More importantly, this approach works with the ocean's natural processes rather than fighting them. We're not imposing our vision of what a reef should be. We're giving larvae the opportunity to build the reef themselves, allowing them to self-organize into whatever community structure works in current conditions. The truly revolutionary aspect is that we're learning to speak the larvae's language. Remember those chemical signals we discussed, the calcium gradients and algal exudates that larvae use to find settlement sites? Researchers have identified many of these compounds and are now using them to guide larval settlement. By applying specific chemicals to degraded reef substrate, scientists can make that substrate attractive to larvae, essentially advertising it as suitable habitat. In some experiments, treated substrate receives larval settlement rates 10 to 20 times higher than untreated controls. We're hacking the chemical communication system that larvae have been using for millions of years, inserting our own messages into their navigation network. Does this sound like manipulation? Perhaps. But consider what we're actually doing. We're telling larvae where they might successfully settle, providing information about habitat quality that they can't assess themselves from a distance. The larvae still make the final decision. They still evaluate the substrate, test it with their sensory organs, and choose whether to commit to metamorphosis. We're not controlling them. We're communicating with them, offering suggestions, creating opportunities. And they're responding, building ecosystems in places where natural recruitment had failed. This collaboration between human knowledge and larval biology is revealing something profound about how ocean ecosystems actually work. They're not static structures maintained by adult organisms. They're dynamic assemblages constantly refreshed by larval input, always just a few generations away from complete reorganization. The adults we see when we dive on a reef are temporary expressions of decisions made by larvae months or years earlier. Change the larval supply and you change the ecosystem. Disrupt larval settlement, and the ecosystem collapses regardless of how healthy the adults appear. Enhance larval recruitment, and degraded ecosystems can recover faster than anyone predicted. This perspective inverts our traditional understanding of ecosystem management. We've been focusing on protecting adults, limiting fishing pressure, reducing pollution exposure for settled populations. All of that matters. But if we ignore the larval supply, we're managing only half the ecosystem. The children of the currents are the renewable resource that keeps everything else alive. Protect them, understand them, work with their natural behaviors, and the ocean retains its capacity to heal itself. Ignore them, and no amount of adult protection will prevent ecosystem collapse. Some of the most exciting research involves predicting where larvae will settle before they do it. By combining ocean current models with genetic tracking of larval sources, scientists can now forecast larval supply to specific reefs with surprising accuracy. They can identify which reefs are larval sources, producing more larvae than they receive, and which are sinks dependent on larval input from elsewhere. This knowledge is revolutionizing marine protected area design. Instead of randomly protecting scattered reef patches, we can protect source populations, the critical nodes in the larval supply network that sustain downstream ecosystems. Protect a source reef, and you're protecting dozens of sink reefs that receive its larvae. It's ecosystem management at the network level, treating the ocean as a connected system rather than a collection of isolated sites. And it's only possible because we finally understand that larvae are not just drifting randomly. They're navigating predictably through current networks, connecting distant ecosystems through invisible filaments of biological potential. We can map those connections, model those flows, and design protection strategies that work with larval transport rather than ignoring it. But perhaps the most hopeful aspect of this new understanding is what it reveals about the ocean's resilience. Yes, we've disrupted larval navigation with pollution and climate change. Yes, we've accelerated invasions and forced species through evolutionary bottlenecks. Yes, ecosystems are transforming in ways that seem chaotic and destructive. But the fundamental mechanism of ocean life, that ancient system of larval dispersal and settlement, is still functioning. Larvae are still navigating. They're still finding suitable habitat, or making unsuitable habitat work through adaptation. They're still connecting populations across distances, still maintaining genetic diversity through long-distance exchange, still building new ecosystems wherever conditions allow. The ocean hasn't lost its ability to regenerate. It's regenerating into something different, something adapted to warmer temperatures and altered chemistry, something we might not recognize or value as much as what existed before. But the process itself continues. The children of the currents are still out there, billions upon billions of them, making microscopic decisions that add up to macroscopic ecosystem structure. That capacity for self-organization, for emergent complexity arising from simple individual behaviors, that's the ocean's true resilience. Not the ability to resist change, but the ability to reorganize after change, to build new structures from chaos. This realization should fundamentally change how we think about ocean conservation. We can't freeze ecosystems in their current state. We never could, really, but climate change has made that impossibility undeniable. The ocean is going to transform whether we want it to or not. Species distributions will shift. Novel communities will assemble. Some ecosystems will collapse and new ones will emerge. But we have more agency in this transformation than we thought. By understanding larval behavior, by working with their navigation systems, by protecting source populations and maintaining connectivity networks, we can influence which species thrive and where new ecosystems form. We can make degraded habitats attractive to larvae again, can guide reef development in ways that maximize biodiversity and ecosystem function. We can even, potentially, help climate-adapted larvae from southern populations colonize northern reefs before those reefs bleach and die, essentially assisting the poleward migration that's already happening naturally. This isn't playing God. It's working with the ocean's own mechanisms of adaptation and dispersal, the same mechanisms that built every ecosystem currently existing. We're just doing it consciously, with intent, trying to steer transformation toward outcomes that preserve as much biodiversity and ecosystem function as possible. So return with me to that image from the beginning. You're standing at the ocean's edge, looking at water that appears empty but actually teems with invisible life. Those billions of larvae drifting through the currents, they're not just surviving in a deteriorating environment. They're actively responding to change, evolving strategies we barely understand, navigating by senses we can't replicate, making decisions that will determine which ecosystems exist 10 years from now, 50 years, a century. They're not victims. They're participants in the greatest reorganization of ocean life since the last ice age, possibly the greatest since much deeper time. And we're participants too, whether we choose to be or not. Our emissions are rewriting their chemical navigation cues. Our ships are extending their transport networks. Our coastal development is eliminating their settlement sites while creating new artificial habitats. Every action we take sends ripples through the larval realm, altering the decisions billions of nearly invisible navigators make every day. But now, finally, we're beginning to understand this. We're learning to see the invisible, to hear the chemical conversations, to map the connections that hold marine ecosystems together. We're learning to speak with the children of the currents, and they're teaching us that the ocean isn't dying. It's transforming. And if we're wise enough to listen, to work with larval biology rather than against it, we might help guide that transformation toward a future that's different from the past but still wondrous, still alive, still full of mysteries we've barely begun to comprehend. The larvae are writing the ocean's future right now, in real time, in invisible ink that we're only learning to read. And the story they're writing is still being composed, still open to influence, still full of possibility. That's not horror. That's hope.